Hello everyone, welcome to the video class on lipids, and in this lipids I am going to explain more about fatty acids. In this video I am going to explain the definition of lipids, lipids biological significance and their classification, and then fatty acids, saturated and unsaturated, omega fatty acids and their significance, and then essential fatty acids. This is my channel. You just type in my name G Sai Rajesh in YouTube, you will get the channel. I have 1840 subscribers and 119 videos related to pharma are uploaded, and my channel has crossed 45,000 views. Let's get into the class. What are lipids? Now lipids are chemically diverse group of compounds. In biomolecules we have carbohydrates, proteins, lipids and nucleic acids. Leaving lipids, all of them has got a similar pattern of chemical characteristics. In case of carbohydrates, they have polyhydroxy group with aldehyde or ketone. In case of proteins, they are made up of amino acids. Whereas lipids, there are very diverse chemical compounds. So the definition is given with respect to their solubility. All of them are insoluble in water. This is what is the common feature. So they are diverse, they are insoluble in water, but they are soluble in organic solvents like ether, benzene, etc. Now solubility in water is because of polarity. Polarity means the ability to generate charge. Take for the example of hydrochloric acid. Now chlorine has got more electronegativity, so these electrons are pulled towards chlorine. So when electrons goes towards chlorine, it acquires a negative charge, whereas hydrogen acquires a positive charge. The ability to generate this kind of charge is known as polarity, like poles. So anything which has got this kind of polarity will dissolve in water. Whereas all the lipids are insoluble in water, they are not getting dissolved in water. The reason is they have higher number of hydrocarbon chains. Hydrocarbon chains means they are all made up of carbon and hydrogen. When you see the electronegativity differences, both of them has got similar electronegativity differences, so electrons will not move to a particular atom. So there is no generation of charges like this. Hence they are not polar, that means they are non-polar. Because they do not generate polarity, they are not soluble in water. This is what is the reason for their insolubility. But they are soluble in organic solvents. Moving to the next one. Now, lipids function or biological significance. Bile acids are steroids that help in digestion of fat and nutrient absorption. See, excuse me, these bile acids are stored in gallbladder. So whenever we take fatty foods, the bile is released into the intestine and they emulsify fats. Basically, they will emulsify fats, and lipase, lipid digesting enzymes, can act on this emulsified fats. So they help in the digestion of fats. Next one, cholesterol is also a lipid, and it is a major component of cell membrane, and it is the precursor for other steroids like estrogens, progesterone and testosterone. Eicosanoids are 20 carbon containing molecules. They act as chemical messengers between cells like prostaglandins, leukotrienes, thromboxanes. All of them are essential chemical mediators. Next kind of lipids, fat soluble vitamins like A, D, E, K, they have multiple functions like blood clotting, vision, calcium absorption. Now this video topic, fatty acids act as precursor of triglycerides, and they are the major source of energy. Most of the animal cells has got triglycerides act as source of energy. Next one, phospholipids. Phospholipids are major components of cell membranes, and they also help in fat digestion. Next one, steroidal hormones. Again, they are chemical messengers between cells. Triglycerides, as I told you, they are energy storage forms. They cause thermal insulation. Most of the animals has got subcutaneous fat. Subcutaneous means under the skin. We all have under the skin certain amount of fat is there, and that gives us a kind of thermal insulation. Temperature control can be achieved with the help of subcutaneous fat. They fill the space and they bind the organs together, and they cause padding or cushioning of organs. The internal organs are surrounded with a kind of fatty layer which will cause cushioning to that particular organ. So all these are biological significance or functions of lipids. All these are various types of lipids. Now in this class we will see more about fatty acids. Now in classification, when you see the classification, They can be classified as simple lipids, compound lipids, derived lipids. Simple lipids are basically esters of fatty acids with alcohol. Ester is formed when a acid combined with alcohol. Now this acid is fatty acid in case of simple lipids. Now in this you have fats, oils and waxes. In fats and oils, the alcohol is glycerol. Only the difference is fats are solids at room temperature, whereas oils are liquids at room temperature. Whereas in case of waxes, they have long chain fatty acid and has alcohol other than glycerol. So all the simple lipids are basically, they are esters. Esters are formed with the combination of acid and alcohol, right? Now in case of fats and oils, this alcohol is glycerol and these are known as fatty acids. Whereas in case of waxes, other than glycerol, alcohol will be there. So these are simple lipids. Now coming to the compound lipids, they have two components, a lipid part and a non-lipid part. For example, phospholipids, a lipid part is there and phosphate is there. Glycolipids, glyco means carbohydrate, a carbohydrate part is there, lipid is there. Lipoproteins, lipids and protein combination. Sulfolipids, sulfated carbohydrate and a lipid. The last one, derived lipids, these are derived from simple and compound lipids. Fatty acids, steroids, glycerol, prostaglandins, leukotrienes, terpenes, monoacyl glycerol, diacyl glycerol, fat soluble vitamins, ketone bodies, a lot of derived lipids are there. So this is a brief classification of lipids. Now coming to the topic, fatty acids. Fatty acids are carboxylic acids with hydrocarbon chains ranging from 4 to 36. The simplest fatty acid is, see, it starts with a minimum 4 carbons. So what is this? 4 carbons are there and acid is there, so this is called as butyric acid. So this is a simple fatty acid. Now you happen to see quite a lot this word, free fatty acid. Unesterified fatty acid with a free carboxylate group is known as free fatty acid. Now see, there is something called as triglycerides. Triglycerides, you have glycerol, alcohol is esterified with fatty acids. Usually in the body, the major storage fuel is triglycerides. Always the glycerol is, alcohol is esterified with these fatty acids. When it is hydrolyzed and it releases this fatty acid, that particular fatty acid is known as free fatty acid because it is not esterified. No, unesterified one is known as free fatty acid. Now the significance of fatty acid is oxidation of this free fatty acid yields much more energy. That is the reason why lipids are known as storage form of energy and major source for lot of energy. So this is about fatty acids. Now when you see the classification of fatty acids, it is based on degree of saturation. When carbon-carbon do not have a double bond, they are known as saturated fatty acids. All the carbons are with only single bonds, like Lauric acid, Myristic acid, Palmitic, Stearic acid. Unsaturated fatty acid. When they have a carbon-carbon double bond, this is known as unsaturation. Mono means only one unsaturated bond is there in an entire compound. They are known as MUFAs, mono unsaturated fatty acid. Example, palmitoleic and oleic acid. The next one, polyunsaturated fatty acid. When there is more than one carbon double bond, they are called as polyunsaturated fatty acids, also known as PUFA, P-U-F-A, polyunsaturated fatty acid. Example, linolenic acid, linolenic acid. You can see there are 2 double bonds are there in linolenic acid, whereas 3 double bonds are there in linolenic acid. Linoleic, linolenic, only N alphabet is different. Now after this, see, these are the examples for saturated fatty acid. Now you need to understand this chemical notation. See, the first one, 12, indicates number of carbons. After a colon, the digit indicates how many double bonds are present. If there is no double bond, it is indicated as 0. If there is one double bond, it is indicated as 1. So this says 12 carbon containing fatty acid with no double bonds, because they are all saturated fatty acids, no double bond will be there. So this is the structure. Look at this. The functional group carboxylic acid carbon will be 1 plus 1, 10, 11, and this carbon is another one. So totally 12 carbons are there. Now the systemic name is dodecanoic acid. Deca means 10, do means 2, oic acid indicates carboxylic acid group. So 12 carbon containing carboxylic acid, saturated fatty acid is known as dodecanoic, DECA. If it is a double bond, instead of A you will get E, decanoic. We'll see it later. So the common name is lauric acid because it is from Laurus plant. Whereas 14 carbon containing without any double bonds or saturated fatty acid is Myristic acid because it is obtained from Myristica fragrans. Tetra means 4, deca means 10, 14 carbon. It follows the same thing. 16 carbon without any double bond is palmitic acid. Hexa means 6 plus 10. It is obtained from palm tea, hence it is known as palmitic acid. 18 carbon one, octadecanoic, 8 plus 10, 18. It is known as stearic acid. In Greek, steer means hard fat. 20 carbon containing one is known as eicosanoic acid. Example, arachidic acid. Arachis, the groundnut belongs to this genus, Arachis hyposea. So all these are example for saturated fatty acid, but out of them, the widely present saturated fatty acids are 16 and 18 carbon containing, means palmitic and stearic are very commonly found. Now after that, unsaturated fatty acid. Look at this. So after this column, the initial one indicates number of carbons and the next one indicates unsaturation. So 16 carbon containing one unsaturated bond. Excuse me. In the bracket there is a delta. The superscript indicates where is the double bond is present. It is present between 9 and 10 carbon, so it is given as 9. Now look at this. The common name is palmitoleic acid. Systemic name is hexadecenoic. Hexa means 6, deca means 10. Enoic acid means a double bond is there. Where is it? It is at 9th carbon. So palmitoleic acid is this one. See, this is how it is written. This is the first carbon and then you have 7 carbons, 7 plus 1, 8. So this is the 9th carbon. So there is a double bond between 9 and 10 carbons. That is palmitoleic acid. Similarly, 18 carbon containing with one double bond is oleic acid. Cis 9, octa 8, deca plus 10, enoic, a double bond. So similarly, the structure is written like this. Now after that, 18 carbon with 2 double bonds is known as linoleic acid. Linoleic acid, because 2 double bonds are there, look at this, octa means 8, deca means 10, dienoic acid, that means 2 double bonds are there. This is how the systemic name is given. Whereas 18 carbon with 3 double bonds, alpha linolenic acid, see octa again 8, deca 10, trienoic, 3 double bonds are there. And then 20 carbon containing is known as arachidonic acid with 4 double bonds between 5, 6, 8, 9, 11, 12 and 14. Just go through the slide again, you will get a clear understanding about how the name is given and how it can be written. To give you examples, see unsaturated fatty acids, see oleic, linolenic and linolenic, all of them contain 18 carbons. 18 carbons, the numbering starts from here, first one, 2, 3, 4, 5, 6, 7, 8, this is 9, 1, between 9 and 10, there is a double bond is there. So C18, delta 9, that is oleic acid. Linoleic acid, again 18 carbons are there. So this is 9 and 10 and then 12 and 13. So delta 9, 12. Linolenic acid, same 18 carbon, but you have 9, 10, 12, 13, 15, 16. So delta 9, 12, 15. Double bonds are present. So this is how it is written. Moving to the next one. See, all of them you can see in the name it is given as cis. All of naturally occurring unsaturated fatty acids are in cis conformation. Cis means similar groups will be on the same side. So this is how it is there. So they are called as cis fatty acids. Moving to the next one. Now omega fatty acids. How this nomenclature is given? Usually in IUPAC systemic nomenclature, the carboxylic acid will get the first one and the last carbon is this methyl carbon. In Greek nomenclature, omega is the last alphabet. So when you say omega fatty acids, this methyl group is known as omega end and the numbering is given from this end. So what happens is, now look at this. This is something called as omega-6 fatty acid. Usually the numbering starts from carboxylic end, but in omega nomenclature, the last methyl carbon is omega carbon. So this one becomes first one. So this is first carbon, and then second, third, fourth, fifth. At sixth carbon, it has got a unsaturation. So because the unsaturation is at 6, it is known as omega-6 fatty acid. Linoleic acid is a omega-6 fatty acid. Omega nomenclature starts from the opposite end of carboxylic carbon. Similarly, omega-3 fatty acid, so this end is omega carbon. So this is 1, 2. At the third carbon you have unsaturation, hence it is known as omega-3 fatty acid. Example is linolenic acid. So this is what is omega nomenclature is. You have omega-6 fatty acids and omega-3 fatty acids are there. Getting into more detail, see, alpha linolenic acid, Eicosapentenoic acid, Docosahexenoic acid. All of them are known as omega-3 fatty acid. Why? From this end, at third carbon, you see unsaturation. So all of them are known as omega-3 fatty acids. Whereas linolenic acid, gamma linolenic acid, and arachidonic acid. So all of these three, linoleic, gamma linoleic, arachidonic acid, are known as omega-6 fatty acid. The reason is from this end at sixth carbon you have unsaturation. So they are known as omega-6 fatty acids. Now there is a significance for this nomenclature. They play omega-6 and omega-3 fatty acid plays a major role in our health. For example, according to American Heart Association, the ratio between omega-6 to omega-3 fatty acid should be between 1:1 to 4:1. That means maximum you can have 4 times of omega-6 fatty acids to, omega-6 fatty acids to omega-3 fatty acids. Look at this. The optimal dietary ratio of omega-6 to omega-3 polyunsaturated fatty acid should be between 1:1 to 4:1. So when you see the dietary intakes of omega-6 and omega-3, it should be either 1:1 or 4:1. But unfortunately, our diets contains 20:1% of these fatty acids. We all take a lot of omega-6 fatty acids because the sources of omega-6 fatty acid is sunflower oil, which we will take very regularly. So there is no deficiency of omega-6 fatty acids, but the problem comes with decreased intake of omega-3 fatty acid. Increased levels of omega-3 fatty acids found to be very protective for cardiovascular health, and they also reduce inflammation. So omega-3 fatty acids are very essential. You can see this slide. See, the benefits of omega-3 fatty acids is they lowers the risk of heart attack, helps asthma patients, reduces inflammation. Both of them are related to inflammation. Rheumatoid arthritis reduces risk of cancer, protects against Alzheimer's and dementia, lowers BP, improves brain memory, raises HDL, which is good cholesterol, and prevent macular degeneration. All these are major benefits of omega-3 fatty acids. And from where we can get omega-3 fatty acids? Fish or seafood, walnuts, flaxseed, walnut oil and flaxseed oil, and canola oil. So increasing the intake of omega-3 fatty acid is very much required. Moving to the next one. Now the last part is essential fatty acids. The fatty acids which must be supplied through diet or which cannot be synthesized in human body are known as essential fatty acid. In humans we have 2 essential fatty acids, are the linoleic acid, alpha linolenic acid. If you remember, see, both of them contain 18 carbon containing fatty acid, whereas in case of linoleic acid there are 2 double bonds are there, whereas in linolenic 3 double bonds are there. Where it is present 9 and 12, whereas here it is 9, 12 and 15. If you remember, this one is omega-3 fatty acid and this one omega-6 fatty acid. Now arachidonic acid, a fatty acid, important one, that can be made from linoleic acid, whereas Eicosapentenoic acid and Docosahexenoic acid can be made from alpha linolenic acids. So remember this, these 2 are essential fatty acids. You can see it here. See, when you see linoleic acid, in its ionized form it is known as linoleate, whereas linolenic acid in its ionized form it is known as alpha linolenate. Now this, see, linoleate can be converted into linolenate in plants, but in humans we need both of them in diet. That's why they are known as essential fatty acids. Now alpha linolenic acid synthesizes other polyunsaturated fatty acids like Eicosapentenoic acid and Docosahexenoic acids. These are required for our essential health. Whereas linolenic gives arachidonate. From these arachidonates we get prostaglandins, leukotrienes and thromboxanes, which are essential chemical mediators. So both of them are called essential fatty acids. Without them we will not get these two. So this is the concept of essential fatty acids. This is about lipids and fatty acids. Thank you for watching this video.